Hey guys, it's Tiffany Martin with Medical Microbiology, and we are in Chapter 7, Microbial Genetics, and we are talking about DNA structure, kind of part 2. We're going to continue on from the previous video. Let's look at this double helix a little more magnified so we can see what's going on here. All right, so there is our double helix, and we have our sugar phosphate backbone. Remember that alternating sugar and phosphates, they kind of refer to that as a sugar phosphate backbone, and then our nitrogenous base pairs across. And so when we talked about pairing, do remember that this is just a shorthand diagram of actual chemical molecules. So there's carbons and nitrogens and oxygens and hydrogens, and they make this up. So if you go back to chemistry, you've got these molecules, and they're made up of these different elements, and they form the specific three-dimensional shape. So when we talk about A's and T's pairing up, it's because of the chemistry, how the different molecules, the hydrogens are put forth. Adenosine and thymine are able to make two hydrogen bonds together, and that's how they're able to pair. Guanine and cytosine, they're shaped a little different, and when they look out, they have the space and the ability to form three hydrogen bonds. So this is why guanine can't pair with thymine or adenine. It just is not shaped correctly. And that's back to its chemical structure. So do remember that all of these shorthand diagrams that we're doing is based on a actual chemical 3D molecule and how it fits together with other molecules. Now my sugar phosphate backbone, this is made up of that deoxyribose sugar, because it's DNA, and that phosphate group, and these form covalent bonds. Remember, they share electrons, and a covalent bond is very strong. Comparison, hydrogen bonds, kind of weak. Hydrogen bonds are very weak interactions, and they're not near as strong as covalent bonds. Now, we said that this double helix is a very stable structure, and that's true. This covalent backbone makes it very stable. And one hydrogen bond, yes, is very weak and can be easily broken. But when you start linking up all of these hydrogen bonds in a row, it ends up being a very strong structure. And you can compare that to a toothpick. I can take one toothpick, one hydrogen bond, I can snap that sucker really easy. But if I put a whole handful of toothpicks together, and I've got like 10 in my hands, and I try to break all 10 of them together, much more difficult. So these hydrogen bonds individually are very weak, but when they form this double helix, it ends up being a very strong and stable structure, which is what you want from your instruction manual. All right, so as for the test, what I need you to know, I do not need you to memorize this kind of stuff right here. Again, if you know you've got biochemistry in your future, organic chemistry in the future, you may want to pay a lot more attention to it. But for purposes of this course and the vast majority of you, just know that this is based on the chemical structure, but we're talking about the shorthand. Now, this is one shorthand version here, this double helix ladder. But then what I do think you should be responsible for in this class is this type of diagram. So how the sugar phosphate backbone is put together, how it is attached to a nitrogenous base. And so that is what we're talking about with this sugar phosphate backbone. So this is sugar is has 6 carbons in it. And these, I think I'm going to switch to my diagram here. In fact, I'm going to start over from scratch so that you can see it drawn out. So it is a deoxyribose sugar, has 6 carbons and an oxygen. And when we draw it out, we have our oxygen. And then every time we get a bend, we get a carbon. So that's a shorthand version for a carbon. So there's 1, 2, 3, 4, 5 carbons. Oh, did I say 6? It's not a hexose, it's a pentose sugar. And there's 5 carbons. 1, 2, 3, 4, 5. Yes, 5 carbons in this molecule for that. And then that deoxyribose sugar is linked to a phosphate group, and you saw that. So I do think you should be able to figure out this type of shorthand and do that. So these are my carbons. So all of these bends are important. 1, 2, 3, 4. This is the 5th one, and so this bend in my shorthand is important because we know that that represents a carbon right here. So that is my carbon for that. Now, in chemistry, they like to number the carbons so that they can keep track of them and what's attached to the carbons. So this is always the 1' carbon there. So next to the oxygen to that is that 1' carbon. So that is always known as the 1' carbon. And off of the 1' carbon in our DNA double helix is our nitrogenous base. So off of this 1' carbon always is where the base fits in and attaches there. Could be guanine, could be cytosine, all of them. They all fit right off this 1' carbon. So we'll continue labeling as we go around. That makes this the 2' carbon, the 3', 4', and 5' carbon. 1 has the nitrogenous base, 2', 3'. The 3' carbon, if I'm just talking about one nucleotide, and the nucleotide phosphate sugar base is a nucleotide. That's a building block for that. Off of this 3' carbon is a hydroxyl group, OH, a hydroxyl group. This is one nucleotide. There's my 4' carbon. Off of my 5' carbon is always a phosphate. That's part of how the nucleotide structure is. So I do think you should know this type of structure. So if I go back to my chart here, you can see there's my deoxyribose sugar, there's my 1', 2', 3', 4', 5' carbon attached to my phosphate group. And you can see how that happens. All right, there's my 1' carbon and it's attached to my nitrogenous base from that. All right, so if I were to ask you to link one nucleotide with another, so if I want you to make this sugar phosphate backbone, how do we do that? And there's a specific way you have to do that because these enzymes are doing the work. And so these enzymes are designed to only do the work a certain way. So when this enzyme comes in to link two nucleotides together and catalyze this building anabolic reaction, it's going to come in and it's always going to look for this 3' hydroxyl. And again, that's just the way the tool is made. It's how it fits together chemistry kind of wise. So it's always going to take this 3' hydroxyl and it's going to rearrange the molecules there and link it with another one. So there's my 5' phosphate. There's my 3'. It's rearranged that hydroxyl group, attached it to the next nucleotide, which has that phosphate from that 5', and then it's my deoxyribose sugar, and then it's going to have that 1' carbon, and it's going to have a base coming out of it, whichever one it is. I call this one G, but whatever it is. What comes off, that's my 1' carbon. What's coming off my 3' carbon? A hydroxyl group. Okay, so if it's going to link it in, it's always going to link it in from this 5'. The tool is not designed to add it to this 5' phosphate. It can never add a nucleotide in on this side. It always has to add it in on this side. And that's going to be important as we start talking about replication. All right, so we just added a nucleotide to my sugar phosphate backbone. All right, so going back to my picture, and looking at that, you can see we've added quite a few nucleotides on this one side, this one backbone. And at the 5' end, I have a phosphate group. And at my 3' end, I have an hydroxyl. And we talked about DNA being 3' to 5'. You've heard that before. This is why. The 5', 3', it's referencing these carbons. And this is the 5' carbon end. So we refer to this as the 5' end of this strand of DNA, and this is the 3' end because it's nearest the 3' hydroxyl. Okay, so 5' to 3', but DNA is a double helix, right? I've got two of these put together, linked by these nitrogenous bases, and those bases are linked by hydrogen bonds. So my first one here is an adenine, and it's got an A, and we know that adenine is going to base pair with thymine. So if I ask you to draw the complementary strand, I'm asking for you to draw the one in blue. So this is the link, that single link, but if I want you, so if I'm talking about linking nucleotides, this is what we're doing. If I want you to draw the complementary strand, I want you to draw this blue strand. And what you immediately notice is it's upside down, right? Again, how that chemistry puts together, it ends up with the deoxyribose going upside down for that. So my adenine binds with my thymine with 2 hydrogen bonds, and we shorthand that with 2 dots for hydrogen bonds. Guanine and cytosine, remember, does 3 hydrogen bonds, so the shorthand for that is 3 dots. And so if I ask you to draw this, you're basically drawing it upside down. So which end is the 3' end? Here's my 3' end, and here's my 5' end. So 5' to 3' here, 3' to 5'. This is that statement where we said DNA runs 5' to 3' and it runs anti-parallel. The strands are running parallel to one another, but because that 3' and 5' is opposite, it is referred to as anti-parallel. And you get the 5' and the 3', and you get that kind of structure. So that's what we're talking about when we're talking about anti-parallel. And that's going to make a big difference when we get into replication as well. Okay, so this is our shorthand. Know what a nucleotide is. Let's go to the next one. Point out some things here. We can see the base pairing and the adenine and thymine. You can see the 2 hydrogen bonds. Guanine and cytosine has 3 hydrogen bonds. Do note that RNA, and many of you already know this, RNA, it doesn't use thymine, it uses uracil. So RNA with this ribose sugar versus DNA and the deoxyribose sugar uses a slightly different one called uracil. And if you look at that, it's almost the same. And so therefore it fits very well with adenine and pairs very well. So it's just a difference between RNA and DNA. Again, shorthand. Oh, the other thing I wanted to point out on this one is I've emphasized a nucleotide is a phosphate, a sugar in the base, and that is a building block, a nucleotide. And sometimes you'll hear a nucleoside, and that's sugar and base without the phosphate group. So sometimes you'll see that as reference. So I wanted to point that out as well. Okay, so here is a time for you to practice. I want you to draw a nucleotide and label it. Seriously, humor me. Do this exercise. Do it with your group. Figure it out. Actually draw it, because when you can draw it, then you fully understand it. Okay, so draw a nucleotide and then label it the sugar, phosphate, and backbone from there. Okay, how'd you do? So hopefully your nucleotide looked a lot like my original one, the phosphate, the sugar, and the base in there with that 3' hydroxyl. Don't forget that, it's going to be important. So that would be a nucleotide. The next instructions tell you to draw 3 of those in a vertical line and link them together via the 3' and 5' end. Don't forget to go into the 3' end of that and do it. All right, how'd you do on that one? I only did 2, but if you saw, we went into that 3' end, not the phosphate. Just because there's room on my board doesn't mean the enzyme works that way. I added it to the 3' end and linked 2 nucleotides vertically for that. All right, do the next one. Next one is going to say, draw the complement. What was the complement? All right, do that one. All right, how did you do? So the complement is going to be the other side. And then you had to draw it upside down. If you would. Sorry, I did that very quickly. I should have done that. Anyway, draw that very good. A and T, how many hydrogen bonds do I have there? 2, so I put that shorthand in. You can see that that went opposite, anti-parallel. So if I were to label this, this would be my 5' end. This is my 3' end. If I were to label this side, here's my 3' end, and here's my 5' end. Okay, so in this case, when I'm going to replicate, I'm going to have to add to this end and go forward. So that's how that works. All right, so the complement and how that drew. All right, so if you have any questions, work on that, work with your friends. If you need to talk to me and we can do personal Zooms, just don't let this go by, okay? Figure it out, take the time. I think that's it. I wanted to stop there. Yes, we'll pick up with the next video on the next one. Keep going. Thanks, guys.